Okay, is it recording? Yes. Okay. Welcome everyone. So I think the plan today was to use some of the dynamical systems ideas that we've already talked about, basically attractors, bifurcations, things like the phase plane analysis, to talk about some stuff that we often work with in neuroscience, and specifically a lot of us who were in Steve Grossberg's used to exist department. We used to use all this stuff regularly without actually looking at it from a phase plane perspective. So I'm guessing you might have just taken a quick look at these papers, but let me just sort of look at them. So the UMAP paper was in our syllabus, and it's Steve's attempt to be to appeal to undergraduates, or at least explain something to them. It's sort of okay that in that respect, but I think it would be interesting to kind of step back and look at where Steve sits in this broader scheme of computational neuroscience, and why he's sort of an outlier, and sort of why those of us who studied in his department are also outliers. I think the best way to think about it is that he's sort of a neuroscientist, but not quite. And he's sort of a cognitive scientist and not quite. So a lot of the way he thought was influenced by intuitions about visual perception to begin with, but also about just intuition about differential equations. So a lot of the more, what in my opinion, successful stuff that Grossberg did was to do with visual processing, object recognition, boundary detection, things like that, which involved to some extent introspection, to some extent mathematical intuition, and to some extent dealing with visual illusions and patterns that aren't really what a sort of standard neuroscientist would study, because you can't really study illusions in animals, and you can't record from a human while they're looking at an illusion, really. Like, you can sort of do that in fMRI, but I don't know what you'll really learn. Given, like, for instance, you guys are probably all familiar with these bistable images, like the duck rabbit or the Necker cube, that can be looked at from two different perspectives. Steve was very interested in that sort of thing, and also in brightness and lightness, and these effects of brightness, and how important contrast is for our perception. So in this UMAP, unfortunately, the diagrams are of no use because they're photocopied, so you can't really see the effect. But if one of you, Karthik, could you like Google and find a version of what some of those brightness effects, and like, so I can just share it. Maybe one of you can just share it in the chat box, because there's lots and lots of websites. Or maybe that Averbeck thing, you know, that, sorry, not Averbeck. There's that 3D chessboard with that gray. Adelson, yes. If you could just find that diagram, that's perfect. So Steve definitely was a neuroscientist in the sense that the way that he wanted to explain things was broadly neural networks. And so that's like sort of constraining your type of explanation to a certain space. And the way I like to think of it is that it is always about local interaction. And when I say local, I don't mean to rule out long distance axons. So a lot of people, they say that the brain has non-local interactions, which I think is just nonsense. Like, I think people should not be saying that, because the effects that are long range with respect to what an axon is doing is still local with respect to the axon terminals. So I really don't like it when people talk about non-local interactions. That should be reserved for, weird effects in physics. Saurabh, how's it going? So local in neural space, which is not necessarily Euclidean space in some trivial way, but it's always local interaction. And since we're dealing with continuous differential equations, we're sort of local in time, in a sense. And so his intuition was, well, we know broadly how neurons fire. They increase when inputs come in and decrease when input goes off. So his Lego blocks were local interactions among these artificial neurons. And the ones that I shared in that Colab Python thing are just sort of building up to that. And so one trick he kind of uses is to say, that these units that he's talking about, these mathematical entities, can be thought of as neurons or groups of neurons. And it sounds a bit like a cop-out when you first come across this, but I think it's reasonably well motivated. And for large numbers of not for all neural phenomena, but for large numbers of them, the average behavior of groups of neurons can be characterized by certain increases and decreases, like increase with input coming in, and sometimes the fall off of firing is pretty rapid compared to what you'll get in a rate model. But if you're thinking about groups of neurons, you generally don't see all of them suddenly shutting off. So there's some motivation there. And if you actually then go and work with, say, spiking neurons and then look at the averages, what you see is often fairly close to what you get from these highly approximated firing rate models. So you have these psychological intuitions and these Lego blocks which he brings together, and that's what this UMAP thing is about. Did you find one here? Okay, I'm going to check. Yes, perfect. Let me zoom in and share that. So, have all of you seen this? But just curious. Awesome. Yeah. So what's striking about these kinds of illusions is that we typically think of square B as lighter than square A. So, but if you would actually take out rip up the image and compare A and B, they're the same pixel value, and there's all sorts of effects like this that effectively show. Here they just sort of directly link them to show it. The crazy thing is that even knowing that the effect is an illusion doesn't really, at least in my experience, doesn't help me not see it. So a lot of what Steve has done in vision is to kind of think about what's happening here and think about how, and there's a sort of underlying assumption which often goes unstated, which is maybe worth stating. The way that Steve approaches perception is that something in the brain is isomorphic to your experience. And what that means is that if I see some shape or some contrast, then there's at a sufficient level of abstraction, maybe something that corresponds geometrically or topologically to the thing that you're experiencing. So when you can describe it spatially in the case of vision, then you expect, or Steve expects, there to be at least at some part of the nervous system something that corresponds to the thing that you're seeing. And what's that? Craig O'Brien corn screech is a good one. That might be, because that's what he's talking about in this thing. So you're seeing this, right? If it works right, you'll see that there's as if one side is darker than the other, but the actual gray level of the pixels shows a scallop right in the middle.
If you scale down, if you scroll down, they'll show that.
This, yes. So the intuition here is that some kind of local contrast is influencing how the overall pattern is being seen. There are a few examples like this, but is this being shared? Can you see this, the Grossberg paper?
Nope.
No. So in this paper, you see that there's depending on what's outside of some color patch, you'll see it as lighter or darker. So this is kind of guiding assumption of, or not an assumption, but the type of phenomenon Steve is particularly interested in, and which you can think about in terms of these neural networks, which they excite each other in local patches. So imagine that you have groups of neurons that are corresponding to different points in the visual field. They can excite each other. So let's say that there's a light patch here. They can excite each other locally, but they can also inhibit in a slightly larger space, which is shown in one of these diagrams here. So if you imagine along the x-axis, this is the strength with which a neuron excites something nearby. That's this smaller, sharper Gaussian. Or it's the strength with which a neuron inhibits another a nearby neuron. So there's wider, weaker inhibition and strong, close inhibition. So you can use these very simple connectivity patterns, which are local, to process edges and contrast. And you can get phenomena that are isomorphic in some sense to what people see. So in this case, for instance, if you can get edge detectors, which are not exactly edge detectors, contrast detectors, which are only sensitive to something that is changing, basically. So if you have a field of a group of neurons that all have this type of connectivity with each other in a spatial dependent way, then the way they respond is only to the regions in the space where there's some step input. So his way of thinking kind of introduces these slightly maybe obscure to a lot. Like if you don't think a lot about illusions, or you don't introspect that much about things like contrast, you might think, well, what does this have to do with anything? But so the way I like to introduce some of this is that there's a lot you can do with competitive dynamics. And so instead of motivating it directly from there, which that's one way you can do it, because those neural phenomena have analogs in just normal computer vision and image processing. Some of you who've worked with Adobe Photoshop and other image processing might have heard of difference of Gaussian filtering. So you can do things where you just select the edges in a black and white image, for instance. And so there are neural analogs to a lot of this. And how many of you would find the statement that edge detection and competition are basically the same phenomenon? How many of you would sort of, find that a thing that you could easily relate to or find coherent? Yes, Karthik, yeah. So that's the kind of the first sort of piece in this way of thinking, is to see how competition and edge detection are similar. And maybe the term or concept that will help link them is the concept of contrast enhancement. That's sort of the common factor between competition and edge detection. So a lot of what Steve's models do in the domain of perception and object recognition and things like that, and boundary detection, is to identify the places of maximum contrast at the appropriate level at which that contrast is most useful for behavior. So they're not just producing things that look like the world for their own sake. The guiding assumption is that when you have neurons that respond to contrasts, whether it's in visual space or auditory space or any other space, or even in reward space, if you think about it, then those can be used and sent to the motor system, where you have a kind of inverted analog of the same kind of thinking. But it's a little harder to get into, and maybe later we can talk about dynamical systems in motor control. But just so that I want us to start really, really trivial with a competitive system and use this phase plane analysis, which we can do in two-dimensional systems with just two neurons. So I guess I'll draw it. Or I already did draw it. One sec. This. Okay, so you see this diagram, right? The So these are really, really abstracted neurons. Just imagine that these circles are neurons, and the X quantity represents their firing rate. And imagine that there's some input, doesn't matter what the input is, but if you like, you can imagine it as evidence for two different courses of action. In this particular network, not much of a network, but basically each cell has its own corresponding input, which you could imagine as like in terms of like a tonotopic map or something like that. Where, like for instance, in auditory cortex, you may have groups of neurons that respond to different frequencies of sound. And the reason that they do that is because of how they receive input from the cochlea. So here, in this system, we just have two neurons, and I have just a feed-forward competition. So these flat lines in a lot of ideally everyone would have a convention, but in a lot of neuroscience we do, where the flat line represents inhibition and the pointy arrow represents excitation. Unfortunately, the color convention isn't used much. So some people use red for excitation, which is just amazing to me. So just think of a network where its own inputs are excited, but also inhibit the competitor. In such a system, which is the very first thing we do, I'll just build up to that. So now we're looking at this. As you see this Python notebook, right? So just to go very, very, in fact, if anyone has questions about the coding part, now's the time, because I'll just go through it. Because it's simple enough that we can really, really understand exactly what every statement means. So consider X to be the activity of a neuron. It could be the firing rate, or you can interpret it as just the number of neurons that are firing as a kind of binary flag. And there are ways to show that these approximations work out reasonably well. And E is the term I'm using for excitation. So imagine a really, really trivial dynamical system like this. You can calculate the fixed point just by looking at it, hopefully. And so when an input comes on, it integrates upwards. And if the input goes off, it just keeps the excitation that it held, because it doesn't have a decay term. That's one way to think about it. So it approaches this fixed point. So think about this. When the excitation is 0, what would you expect the system to do? Yeah, so the derivative is 0, so that means it won't change. But if it already has some level, for instance, because of prior history, it'll just keep that level. So you can look at, with simple differential equations, you can look at it and kind of know everything that there is to know about it, but there aren't that many differential equations that you can do that with. So I say that this is a trivial, perfect working memory kind of system, because whatever a level it attains, it'll basically attain its maximum level if you give the input enough time. So see, there's this transient period here where it's working towards that fixed point. If you don't let it reach there, it won't reach there. It will just stay at whatever level it happened to reach. That doesn't really happen with isolated cells. They need, there are cells that are intrinsically active, but that we can just ignore those for now. So we add this lambda x term, minus lambda x. So again, if you were to ignore the excitation part and just look at minus lambda x, we've already looked at those kinds of systems, exponential decay. It's like first chapter of Strogatz, basically. So, and this is analogous to an RC circuit in electronics with a resistor and a capacitor. If you are familiar with Kirchhoff's laws, you can just write down quickly how an RC circuit works. and you'll find that it has the same functional form as this. So while the charging of the capacitor is happening, you get this shape going up like this. And then if you turn off the applied voltage, the capacitor discharges like that exponentially. So this is a model of frequency response in a neuron which has no interesting connections. So now we'll look at this zero-sum game. Well, the sum is one, but basically, this is the pure feed-forward network that I drew, where the inputs cross each other. Open this. Yeah. So here, EI is the corresponding input, and EJ is the input from the other, the competing, say, plan. So this is like just very simple algebra to show that the final point that you get will be the cell's own input divided by the total input. And this is true regardless of how many cells you have. So even though this is a really simple system, something like this is pretty useful if, for instance, you're interested in something that you know has to sum to a constant value. And what might be, what can you think of a very important quantity that you might want to always sum to a constant value? The Saurabh, you were about to say something?
I was going to say probabilities.
Yes, I'm glad someone said it. But people know how I give Bayesians a hard time. But actually, it's good to be able to keep track of probabilities if you can. And the interesting thing about this equation is that you can set this up so that your excitations don't have to be constants, and you can have stochastic input coming in. They could be discrete quantities or continuously varying quantities. And if you just pick the right time constant, it should integrate slowly, basically, then your activities will track, will basically be an exponential running average framed as a probability. So it's an interesting way to think about what a neural network can do. And this is not some discovery of neuroscientists. This is equivalent kind of differential equations and difference equations are used, for instance, in Kalman filtering, which is a way of online keeping track of averages and also variances, which is very useful for online learning. And by online, I mean like being able to respond in real time to a changing environment. So in this little simulation, I have an initial, and I guess a thing to point out here is that because the excitations are related to each other by this ratio, their absolute values don't matter. It's only their relative proportion that matters, which is exactly what you expect of probabilities. So here I have, the input to the blue neuron is larger than the other one, and so the activity of that one is higher. And when I change the inputs, you switch, and this green is the total. So once the system equilibrates and reaches its level, then the sum remains one. So it's sensitive to changes, but it sort of obeys the probability rules. If things change too fast, then you can get transient periods where things don't sum to one, but that's not, I think, a huge problem for practical applications. So that's a feed-forward network. There's no there's no feedback at all.
One thing before you move on from that,
sure.
I was gonna just throw in another example of where that sort of normalization is useful, and it's in artificial neural networks. This is a step that oftentimes, if you're doing image processing or have a convolutional neural network, you'll manually normalize your image so that it's summed to some constant activity or whatever, but basically in effect manually doing the same step that you're doing or getting out of this equation for free.
Yeah, it's normalization is so important that people keep rediscovering it and giving it a name.
Layer norm, batch norm, what are the different terms for it? They have different, all the silly terms. Yes.
In general, it's much easier to deal with relative comparison than absolute comparison. And when you don't know what the range of the quantities of interest are, or if you know that the range could vary over many orders of magnitude, then you really need to do something that is like this, to refactor in some way and look at relative. And so with those on-center, in spatial domain, you can use like local averages, and that's what Steve does to explain some of those brightness and lightness effects. But okay, so this is just the feed-forward network. And now we come to the, I say the winner-take-all network, but this is you can make it many ways. What we like about this type of network is that it contains a normalization of sorts if you need normalization, and it's continuous, or it can be. So okay, so now already we have a slightly more complicated network. In fact, quite a bit more. So we still have this term which marks out the maximum point. So this whole term is positive, right? When x is less than x max, and when we set it up so that it doesn't really go above that. If it does, it'll come back down if you set everything up right. So you have your external input coming in, and this term is local self-excitation. It's not really clear if an individual neuron excites itself, but if you go back to imagining this as a group of neurons, then almost definitely you have groups in a sufficiently large, say, section of the cortex, you'll have self-excitation. You don't necessarily have that anywhere else in the brain, though. Maybe the amygdala and the hippocampus. Yeah, actually, the hippocampus, yes. So hippocampus, amygdala, and cortex are the places where you expect to find local self-excitation. Karthik, are there any other places where that happens? As far as I know, those are the only ones. So that's very useful when you want the equivalent of amplification. And so this function means that we're not just linearly feeding back x, we're doing something to it. I'll show you what we do. And here we have inhibition and here it's not the external inputs that are doing the inhibition, it's the local neuron. So it's rather than the signals that I receive that are inhibiting my competitors, it's that I am inhibiting my competitors directly. That's the way that this works. And with this X-Men here, what this means is that the system can now be hyperpolarized. It can go below zero, meaning that it's not just not firing, but sort of more negative than it usually is at rest.
Did your bottom-up inhibitory input get dropped here?
Bottom-up inhibitory, yeah, I dropped that here. Right? So right now I only have the excitation.
Would it be possible to draw a simple diagram like you did for
yes.
the very beginning?
Sure. Yes.
Johan, if I can share, I've already drawn it. So.
We've already drawn it. Okay. One sec. Good. How does that work?
I should be able to share now, right? So.
One sec. Okay, now you should be able to share.
Can you see it?
Yes. It's some kind of car.
So this is basically the inhibition. So this is inhibition. So this is excitation. And these are the two cells. And then this one, in this case, I'm just saying X1 is getting an input, but I'll just say E1, E2. So that's basically what that equation that Johan has there, where this f is some monotonic function. That's about it. And we can talk about what that function, signal activation function is.
Yeah.
Any questions, Scale? See it? Okay. I'll stop sharing because the cool ones are all in Johan, the simulations.
So it becomes a tangle even if you have 3 neurons. Like we would have to draw these things occasionally for like a poster or something. It was always a hassle. It looked clean for 2 neurons, and for any other number of neurons, it's a big mess. So the main thing is to understand the 2 neuron kind of version, and then it's just every pairwise relationship is exactly the same. And which it actually is not easy to keep in mind. But that's basically how every single one works. And then when you have distance dependence, it becomes even more complicated. But for now, let's assume that all neurons are equally close to each other. So this is the general form. So basically, what is it saying? It's saying that a neuron can be excited by external input. If it's already active, it can make itself more active, perpetuated. And it is dragged down by its peers. So other neurons, if they're also active, will pull everything down.
Is that a typo there? Should that be gxj minus lambda xj?
Yes, yes.
Okay, cool.
J. Sorry. And it should be j not equal to I. But
Oh, but still lambda xi, I guess, because that's the decay.
yes, that's the decay term.
That's the decay for
And
that.
that's not inside the sum term. Yeah, I don't know.
So you can think of any of Steve's networks or neurons in the network having broadly three parts to them. There is an inherent decay, which is that lambda xi blah 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 term, and then there is a net excitation, which is driven by inputs and whatever the corresponding network neurons contributing to that excitation are. Let's say if it's a spatial system, then you'll have a Gaussian kernel or something like that. And then there is a net inhibition term. So those are the three broad things that Johan has shown here. He's first shown the excitation, then the inhibition, and then the decay. So that will be standard. And if you read any of his papers, you will see that kind of repeat. It's not going to change.
I think it's also interesting to think through the cases of how dx/dt changes based on what happens at those boundary points when you hit x max and x min. All right, so like once your activity x hits x max, your input, your bottom up ability to excite the cell shuts off because x max minus x goes to 0. And you're only left with the inhibitory portions and so on, right? Like the decay portion, once you get to 0, in this case, if x min is 0.
Or x goes to x min minus x min.
Yeah, let's say x hits x min. The ability of other cells to inhibit this cell goes to 0, right? And actually at x min, if x min is not 0, really what you'll get is you'll start increasing back up to 0 from x min, but if x min is 0, you just stay at 0.
Yeah. You won't actually ever reach 0 because it's a continuous system. But okay, so that's the network or the equation, and I just solved it for 2 random inputs. So yeah, in this particular case, what have I done here? I have very, very small differences in input. They're tiny. So in principle, this will amplify any difference in inputs. So it's basically a perfect winner-take-all system. If your initial conditions are such that both the x's are 0, then regardless of how small the input is, it will always pick a winner, meaning that one of them will be at its max level and the other will be low, negative, basically. What's interesting about, that's pretty unrealistic to imagine perfect amplification, but it just shows that these faster than linear when I say faster than linear, I mean just, well, in this case, it is linear, but feedback in general is can amplify anything. So I just plotted this in phase space. So the initial conditions were at 0, 0. And because the x-axis term has slightly more excitation, that's the one that ends up positive and the other one is pretty low. Here, there's a little bit of input, but because the system has already picked a winner, the fact that the second one has higher input is no longer relevant. So as one way of thinking about this is a very stable system. Once, in some ways too stable, because once a winner has been selected, the system is stuck, basically, and you need to add additional machinery to bring everything back down to 0 to do a new competition. It's also way too sensitive to, so basically this amplifies any kind of noise. So a system poised to be a pure faster than linear integrator, competitive system, is like a tripwire that responds to dust, basically. Anything will cause it to blow up. In fact, you don't even need any input. So I'm going to set both of them to 0 and just change the initial conditions. So now x, the second one, will have some non-zero value. Yeah, I think this should work. So you see, we started from the 0, 0 point, and we ended up where the other neuron wins. There was no input. It's just that the initial conditions were such that one of them won. So this is a kind of mutually exclusive activity kind of network. Now, yeah, any questions, or if anyone if you want me to like tweak something in this is, I think, fairly straightforward. So you can actually change the decay rate so that it does actually respond. I'm not sure what value will help, but if you, okay, let me first make it so that, right, so here what happens. So this amplification is not very strong here, but do it this way. Yeah, okay. So here I've changed it a little bit so that there's more decay, and now it sort of faithfully responds to changes in the input. So first one of them is active, and then the other moves in state space. State space is actually changed because the input is changed, but so you can't really call this a state space, but sort of what it is. So that's a basic winner-take-all network, pretty useful for things like, so you generally won't use this in isolation. You'll have some evidence accumulation somewhere, and then the final stage where you want to pick, say, a category to say this is definitely a dog and not a cat or something like that. At that final stage, you'll use something like this as your output, because then downstream that can trigger some behavior or something like that. And you want it so that it's, completely mutually exclusive. So then what I did was, so Kale was talking last time about using phase plane analysis. And since most of the time we deal with high dimensional systems, none of us really have bothered to do this. But then I thought, well, I've never looked at the competitive, the two-neuron system in the phase plane. So the code looks a little longer, but so for the, so just to remind everyone, let's go over one second. The behavior of the network depends very strongly on F and G, even in a two-neuron network. So you can have monotonic functions of different sorts that increase like x square, which is faster than linear, or you could have something that's flattened, or then you have sigmoids, which have both slower than linear and faster than linear parts, and a sort of roughly linear part. And that can be very useful for a lot of applications. Like sigmoids are generally the way to go for a lot of systems. So here I'm looking at
in general, if you want to preserve a pattern and exclude noise in the input, we usually go for a sigmoid because it does amplify the important ranges in the pattern, saturates the very, very high amplitude values that you will find in your input, and usually uniformizes or removes all the low noises that you will see. So if you want to have some kind of a pattern preservation, we go for a sigmoid. And then on top of these pattern preservations, if you kind of want to do categorization, then we'll apply like a winner-take-all on top of that. So that's kind of how usually we do it. Very rarely you would see any of us like applying a slower than linear or a linear system, because it's linear one amplifies noise so crazily. So it's not good if you're having a big network doing a lot of different things. And I mean, at least in vision, we do that. I don't know about other modalities. Yeah, usually restricted to sigmoids and winner-take-all, faster than linear for categorization.
Yeah. So also, like physiologically, you probably want something that's bounded on either end, and that's monotonic usually. But sometimes people do use like piecewise linear though, like which is more or less kind of almost the same thing, but not really. So
yeah, the analysis becomes a little bit more confusing when you have these, sharp like where the derivative isn't defined in some points, but it's, so the point about faster than linear and noise is based on a very simple mathematical fact, which is that, like, if you look at x squared, right? What does x squared do to numbers? Most numbers, it makes them bigger, if you want to think about it that way. But if x is less than 1, x squared makes the number smaller. So when you set up a system with these kinds of nonlinearities, you can set it up so that if you know how big the noise, like if you know what constitutes a small signal, sometimes this can be, just trial and error, then you can adjust it so that only signals above a certain strength can get taken up in the system, and everything else gets squashed. And that becomes really useful for the stability of the system. The opposite, in fact, for a long while, I didn't know why you would want slower than linear, because what that effectively does is kind of reduce differences. It's the opposite of a winner-take-all system. It's kind of like an everybody takes some system. And I've been thinking that it may be something vaguely like that may be useful in contexts where you want to explore or you want to reset, or there's a few things we can, it's all speculative, but we can talk. So we made the phase plane. The diagram that I drew is pretty dense, but I'll try and explain it. So this is a winner-take-all system with slightly quirky functions f and g. But so what I did was, so the basic arrow thing, these magenta arrows is your normal phase plane. So what are we looking at? The minimum to maximum and minimum to maximum of the two neurons. And the green dot is where I place the system to begin with, and the red dot is where it ends up. So if you remember, one way to calculate if there are fixed points is to look at the null claims, which is where the derivatives of the quantities become zero. And if you have a two-dimensional system, you'll have two null claims, and where they intersect, if anywhere, then that means that the system is not moving. The system not moving can be because it's a repeller or an attractor. So in this case, the null claims are these fairly complicated cubic structures because of the form of the overall equation. It's called the shunting equation, slightly obscure reasons, but so what you see here, and so the reason these lines are not exactly lines is because calculating null claims is actually a somewhat non-trivial task, as I discovered by accident on, was it Friday? So I was trying to draw this and I had forgotten how to draw these things. And they're basically something called analytic curves in mathematics, where, so even the equation of a circle or an ellipse is an analytic curve. So if I told you, x squared plus y squared is equal to 1, how would you go about drawing this? What values of x would you choose to draw? People who know how this is done, they'll pick a parameter and they know that you can frame it in terms of cos something, cos of something times t, where t is like like time. So you parameterize it. But for in general, parameterizing it to show the exact value of where the system goes to 0 is I'm not even sure how to do it exactly. But you don't have to do any of that, because I just sort of approximated it. So what I did was I looked at so basically you discretize the space and you compute the derivatives of the two for every point in that discretized mesh. And then I just pick a threshold and say that everything below this particular value, I will plot those. So for places where things are pretty calm, you get something like a line. And in places where there's a lot of sort of change happening, I might pick up a little bit more than what I wanted. But basically these are, so the null cline, this isn't the null cline, it's sort of where the null cline is. It's somewhere in there. So you can see that this is a stable attractor. How I know that is by looking at the arrows and the fact that the null clines meet. If I just saw the null clines and not the arrows, I wouldn't necessarily know anything about it. And this one is also stable. So what are these 2 points? They are the points where neuron 1 is the winner, and the points where neuron 2 is the winner. And what this shows is these are the places where you can set up the system initially, and this is where they'll end up. So here it's right on the middle. Because of the way this is set up, it doesn't really matter what the inputs are, because it'll just amplify everything up to a point. The color is just the absolute value, like the background color, the greenish sort of thing. The contour plot I put on it is just the absolute value of the gradient. So it gives you the how fast things are changing, basically, but doesn't tell you the direction unit is changing. So the direction is given by the arrows, and how fast is given by the color. So things are pretty fast out in the periphery, and then as things reach the fixed point, things slow down.
Would you call the 0, 0 point a saddle point because it's got attractor from 2 directions and then going away in the other 2?
I believe this is a saddle point. Yes. Well, yes, it's a saddle point, because the arrows are coming in from this direction and they're going out from this direction.
That should be like where the inputs are the same, right?
Right. Well, the inputs being the same, just kind of it's hard to understand exactly, meaning I don't have a good intuition for why this is, but it will just squish the system. It'll look exactly the same, but will kind of deform the system. And let me show you that. So right now I actually have that already, but I'm going to make one of them much larger. And you see what happened. Null cline moved. So that isn't even, so now I only have one stable attractor, because the excitation was so much more. So in such a system, no matter what initial condition I put, it will always find its way here. It might sort of briefly find its way here at the other one, and then sink and find its way to the other, to the actual winner. And well, it's still a saddle point, but I actually placed the initial condition right on the diagonal, so it couldn't break symmetry. So now let me move that. Where is it? Yes. So if I just move this slightly off of the diagonal, it should break symmetry. Oh, maybe not. Wait, let me think here. Okay, right now this is the only. Oh, of course, yeah, it's intersecting in only one place. Okay.
So I don't want to distract too much from the awesome, pretty cool diagrams, but just something I mentioned earlier. So that the cells, like I mean, at least by the Hodgkin-Huxley model, and we kind of talked about it a little before, don't really saturate on the upper end, right? So like the rate just gets faster and faster. I mean, it does kind of level off, but it doesn't completely level off. It continues to grow.
It'll just die.
And the oscillations get faster and faster, but this height of the spike gets shorter and shorter
Right.
at some point. And then you might consider it no longer a spike by some arbitrary measure, but it sort of doesn't really reach like a
Yeah, the firing rate, that's true, doesn't
definable threshold. And of course, I mean, a definable plateau. And of course, that also is like well beyond what would normally happen biologically. But yeah,
in those situations, the Hodgkin-Huxley network might not actually, model a cell dying, right? So we actually saw that, the excitation block, when we looked at the Hodgkin and Fitzhugh model.
To Jesse's point, does imagining this as a group of neurons help with that, as this is the firing rate of a region?
It's arbitrary. It's very hard to say what it can be. You can even imagine it as a bunch of, in the simplest sense, just think of it as a bunch of neurons, just a multi-unit activity. So you can get instead of looking at spikes, which is harder even for an electrophysiologist, you can just take the rough average firing rate. So yeah, but
it kind of depends on how you're justifying that sigmoid. I mean, if you're justifying that sigmoid based upon the like how active a cell will be based on its input, then it doesn't, like, the sigmoid doesn't flatten out. It keeps going. So, I mean, I don't know what other way you kind of justify the sigmoid, but it's still a sigmoid. It just doesn't, it just kind of like doesn't quite level out. It just kind of keeps rising at the tail end.
Yeah, but if it's a group of neurons, then there's no nothing can increase to infinity.
And at the same time, you shouldn't be too extensive in or broad in how far you can go with this. You shouldn't just be like, yeah, this is 2 areas, 2 whatever. You don't want to extend it that way either. So there's sort of like it's a sort of coarse graining, but don't take it to be extended to any 2 big systems and put them together. So yeah.
The question, I love this sort of trivial statement, right? It's like, it's true as far as it goes, but the as far as it goes part is not inside dynamical systems theory. That is something completely outside of it. Like, because whenever you make a model, you can study the properties of the model. That's what we're doing here. Not if the model breaks down, when the model breaks down, we won't have any explanation from inside the theory, from inside the math. We'll have to say that the model wasn't good enough. And that's, I think, it's, trivial, but really important to keep that in mind whenever you're doing this, that model will break down eventually.
But in this case, we actually brought it up. I've actually brought it up a couple of weeks ago, because in reference to the Zikovich model, saying that, like, is this observed physiologically, like at high inputs? And we said, yes, it is. But
oh yeah, some of those things
maybe only in slices. So not necessarily in a like in a living organism, but
exponential increase of firing is also rarely observed in vivo. What you often see is a pattern which Karthik probably sees regularly. He records from LIP, and so you see when there's a stimulus-sensitive region, you often see a rapid bump of activity, followed by so a bump, a decrease, and then another gradual increase. I think you probably see this a lot, right?
It varies from region to region. So you'll usually see something like an impulse response once the input comes in. So it's like a blast of input, so the system doesn't really know what's happening. And that going down, the dipping down will kind of vary depending on the region. So it's sort of like harder to say. Like, in fact, you would see that dipping down more as you go further away from the sensory areas. And then it kind of like starts climbing up and like flattening. And if those stimuli were sustained, you will see kind of like a more, let me just say for the moment that it is modulated, but it's mostly flat. And that is the one that most of us are interested in. We don't care about the initial impulse response part, because that's when the animal or the human being is doing some interesting behavior, whether it's attending to a target or holding on to some object in working memory and so on. So we usually neglect the first 150 milliseconds of the signal once the input comes in, because it's not useful for any analysis. It's useful to some extent to just say like how driven a particular neuron is, because the amplitude of it will tell you something about it. But beyond that, it's not really helpful.
It's actually fairly challenging to get spiking neural networks composed of Hodgkin-Huxley neurons to replicate the shapes of these firing patterns. I've always been intrigued by them, but I've never seen a model that really looks like that. It's like if you make a network of Hodgkin-Huxley neurons, it's an incredibly brittle kind of like, especially if you have local feedback, it'll shoot up to the maximum level that it can have, which is way like it'll go to hundreds and hundreds of hertz. If you don't introduce additional currents and interesting ways of preventing that from happening. So there's an interesting, like a lot of people who are aware of these types of equations, which have, yes. So this type of equation, you won't see it in this, but because I showed you the probability thing, there's a normalization that's going on, in this case, online normalization of some form, right? It's very hard to get spiking neuron models to show normalization. So sometimes you'll hear this as a criticism of rate-based models. It's like, oh, I can't get the same effect with a Hodgkin-Huxley or an Issakovich neuron, and I trust the spiking. But the crazy thing is that if you look at actual recordings, you do see normalization in many places.
Not in many places, almost everywhere.
Almost everywhere, exactly.
Almost everywhere in the cortex. So
what this suggests is that even though the Hodgkin-Huxley model is the most successful biological mathematical model ever, it is not adequate for modeling networks. So there's something we're missing. I mean, there are some very, very ad hoc, hacky ways of getting divisive normalization. That's the term. With spiking, they involve making sure that it's very like random input and like fairly low input, not a whole lot of feedback. So there's ways you can do it, but we don't have a very strong principled way of getting this kind of normalization from spiking neurons. So the normalization is there. People observe it in everywhere basically, but modeling it directly involves, there's some emergent phenomenon at the network level that we don't know how to do in a bottom-up way yet.
I don't know exactly what you mean here by normalization.
So this step here, in not so much in this one as getting the firing rate to be divided, basically. They call it gain control, some people. So gain control is something widely observed in the brain. Basically, the slope of the firing response to input changes rather than just shifting the whole slope. You see both in the brain, shifts and gain control. But if you're trying to get gain control with the spiking model, with just excitation, inhibition, and normal, AMPA and MDS synapses, you can get it. There's a bit of alchemy involved in getting it to work.
I think you can do it.
Go ahead and do it.
I mean, I think my model may have been doing that, my paper from before. I mean, it certainly does all the things that that like the winner take all versus the flattening, and the flattening I think normalizes any or the in the linear regime, I think it would
That's cool. So
maintain the pattern and normalize it. I'm pretty sure.
there must be
But it's only
some
going to be within
additional,
a certain range of input.
right. So that's the additional structure, which I suspect has to do with the way that the currents have been set up, and probably also the neuromodulators.
Yeah, I mean,
It's
it certainly.
not like people haven't done it, but
Well, that model doesn't involve neuromodulators. It's just that in the basic version, it's just exactly like this. It's just lateral inhibition and self-excitation. But there are some key things to getting that to work. One of which, well, at least when I did it, which is if it did that, I never really checked to see if it did normalization. But I mean, I think it would in that linear range because it behaves exactly like these shunting networks in at least the other extremes. But the like increasing the capacitance of the cells kind of allows for like them to act more like integrators and be less kind of noisy in terms of their responses.
it's not about integration so much as how the inputs are treated by the neuron. So in a Hodgkin-Huxley or Itzkovich of its Gumo kind of model, the inputs, both excitation and inhibition, are additive. And the relationship between those inputs and the firing rate is that, yeah, the input will increase the firing rate, but the inhibition, all it does is reduce the excitation, right? Like in a standard, even if you have interesting synapses. So those will tend to just shift the whole pattern, basically change the threshold, but have the exact same frequency response. So you need to have something in addition to that, because you'll be able to see it even in the equation, right? That the excitation is just excitation minus inhibition. You'll never get showing the frequency response in functional form is impossible, basically. But yeah, I'd be curious to see it, because
well, in the aggregate of cells, though, you can because you have. I'd have to think about this a little more, but
I've tried it. It's like very hacky.
Yeah, I'm curious. I can't say that.
Yeah,
I've actually done it.
Yeah. It's because you can get winner-take-all networks to work just fine, and if you've decided that your input lives in a particular like single order of magnitude, right, then you won't notice. It's only when you are looking at a couple of different orders of magnitude that it really starts to become useful to have gain control. And most people don't necessarily need to model all of the 2-3 orders of magnitude.
Yeah.
But in vision and hearing, we decibels, we do actually have sensitivity across orders of magnitude. Yep. But yes, there's a lot of interesting stuff to model in that, in just trying to get spikes to do things that rate models do, and which is in the data. So that was this.
My problem with all that is more practical. Like, why would I be using a Hodgkin-Huxley model when I don't see anything even reflected in my the simplest analysis that I as an electrophysiologist does has nothing to do with spiking. So why go through all that effort?
Yeah, what do you mean? Like you record spikes, right?
Yeah, but we can't see anything without spikes. We require some sort of averaging or rate modeling. Rate-based averaging is the only way you will see any of those profiles. And we are not making statements about the brain based on the spikes at all. Almost never.
Yeah, that may be fair. I'm just saying that I'm not sure that the Hodgkin-Huxley is deeply inadequate, though. It just might require the right parameters and the right kind of thing to
people
do.
have done it. It's just that the versions that I've seen, and I've gotten it to work
Yeah.
myself, it's really, really hacky.
Yeah.
Especially if you want it to work for 2 or 3 orders of magnitude.
Is there a theoretical problem here that's take the Hodgkin-Huxley model and then compute get an effective model for just the rate of several coupled Hodgkin-Huxley models? Is that the problem? Somebody solved that.
The frequency responses, but there's so much sensitivity to time that that for relatively sparse inhibitory events that are not synchronized with the excitatory events, you can get many different effects. But yes, I think a good theoretical perspective might help clarify how to answer the questions or come up with effective theories. I'm not sure exactly how to do it, but yes.
You're saying that like say one is driving the other at resonance, then you'll get these kinds of special effects.
At resonance. Well, yeah. So I don't know exactly, but I have tried all this in the past with Itsiguish neurons, and my experience was that if you have highly correlated excitation and inhibition, then this pure additive kind of effect is not always true. So if your inhibition is really well timed, so it comes right after the excitation or right before, then you can get some interesting scaling effects and changes to the firing rate, the response. And
what are the common ways of like modeling postsynaptic potentials, like EPSPs and stuff like that? Like, how do they translate from the firing rate of the presynaptic neuron? What are the common models for that?
let me show you Jesse's model, which is basically this equation.
There are multiple models. Yeah, I actually it's Dysteki at all is actually the model I took and kind of modified it slightly, but Dysteki is, I don't know.
Yeah, I've been citing Jesse.
I don't know. Well, I
Yeah,
dug
because
actually
that might help.
that paper recently, and it's I'll just put it in the chat to where like I actually got that, and it's actually at least roughly based on someone else's model. So
yeah, it works very well, and it's way better than using double exponentials. So should I show it? It's the same as one of these equations we already saw, basically.
There's multiple models is the short answer. Like the basic thing is like a decay is like an impulse and then a decay.
Yes. So it looks like
this is a little bit more complicated than that, but
it's like this. So imagine that your E now is a postsynaptic event which shoots up, and this is the postsynaptic potential. It's the same thing basically. So you have this temporally brief increase in potential, and then it decays. It won't actually reach the max because it's fairly small. So a bunch of them can accumulate basically, and it basically works like this.
Yeah, so it's a leaky integrator of a leaky integrator essentially, where the first one is representing the neurotransmitter and the second one's representing the activation of receptors as a result of the neurotransmitter. That's the model I used. And the initial impulse response is essentially just width of a spike, so it's 1 millisecond. It's just a 1 millisecond input, like this square function.
So the things that Jesse is talking about are the things which end up in the Hodgkin-Huxley network just being two additive terms: the excitatory part minus the inhibitory part. So whatever interesting effects it has on firing rate that aren't just a shift property have to deal with the timing, as far as I understand it, or with some additional effects, nonlinearity is introduced at the time of integration of the arriving postsynaptic potentials, as far as I can tell.
For sure.
But timing can really change how a spiking network works. Like if excitatory and inhibitory events, which are these little punctate events, if they're very correlated, you can get quite interesting, unusual behavior. And there are networks where it does seem EPSPs and IPSPs.
There are issues beyond just timing. I have no clue how I'm going to put up like 100 Hodgkin-Huxley neurons together and do something. So I'm okay with us trying to find a mechanism for how a spiking neuron might do all these things. I think it's a good biophysical idea, but I don't understand why this will help us in any way if you're going to talk about actual big networks. We have to at some level abstract away from whether it is spiking or not. We have to abstract those values, and I don't see a way out.
I think that that's true in general, but there's two reasons why you might want to use spiking. One is STDP, spike timing dependent plasticity. So if you're studying plasticity and you want to use these interesting spike timing based things, and you can't do that with rate models. The other is to please reviewers.
And to like effects of neuromodulators, right? I mean, you can model that with a rate-based model or some aggregate model, but it's sort of more justified in my mind when you have like an underlying physiological mechanism that it maps with. I agree that like large models of Hodgkin-Huxley are probably ridiculous, but there might be times where you want to like kind of match that up with the underlying biophysical models so you can understand like the actions of drugs or whatever.
Yeah, I agree on the part that we need to bridge the scale between the spiking world and the rate world. I agree on that part. But even at the STDP stuff, I am not convinced, because how often do you actually see people recording STDP in vivo?
Almost never.
Okay, so it's all fine in practice to say that this system has these biophysical limits it goes through and all that stuff. Show me the evidence of it when it is actually working, then I'll start taking it more seriously. Because for me, I don't have the luxury as an awake animal recording person to actually like speculate on whether the kind of plasticity that is happening when this animal is learning a task is STDP or not. I have no access to any of the synapses. I have to just look at what the firing rate is and come up with some ways of talking about adaptation and all that.
Right. But the modelers have to go beyond the data, right? Otherwise,
but if you're not going to tell me anything about how it'll relate back to my data, then that modeling is at some level completely distanced from the data that I'm saying. And these are models that are doing the opposite. They're saying that I'll try to at least account for some aspects of the data.
Yeah, I mean, I generally agree. I don't think Hodgkin-Huxley should be used all the time. I'm just saying.
I think that, like, I agree with Jesse that, in fact, I've been using Isikafish models for a lot of things, just because in some cases they're just easier to work with when you're not dealing with many orders of magnitude. I also ran, I just wrote a version of the winner-take-all for higher dimensions, which is what most of us would end up using, or versions of this, where it's not just 2 dimensions. And here we can't really look at the phase plane. And when I was coding this, I just plonked the faster than linear transfer function in, and I gave the system random inputs. And I got an oscillation, which I was not intending to get. But I knew that, and in fact, Himanshu once just popped one of these into his model because he needed it for some reason. But basically, this is a competitive oscillator. It doesn't have to be. It's sensitive to this decay parameter, for instance. If it's less, then it won't be a selector. Well, maybe I have to increase it. But yeah, so you can get some weird things. Well, now it's not competing. But this will basically amplify small differences in the input, and it'll always pick in the right parameter range. It will pick whichever cell has the most input. So you see here. So what I've done here is, so right now the input is constant, some random quantity, and then this is just the sort of continuation. The input changes and it picks a new winner. This is one of the things that I actually think oscillations are good for.
Sorry, Jan, what are the X max, or what are the cell max values? Is this basically, I'm curious if this oscillation's an artifact of like an Euler step being too large.
Yeah, it could be, yeah.
And this is something that bit me in the ass for 3 months of my life, but if it's just stepping over 1 and so creating like a sharp pull downward or whatever, as
right
opposed to
over
like
there.
slowly approaching one.
Meaning like in this downswing.
Yeah, like should the selectivity be capped at 1?
Oh, I've made it 2.
Okay, so it's not an artifact.
Yeah, I'm not doing that.
Okay. Well, because depending on how sensitive the system you've set up is, if your Euler step size is too large, then there's all sorts of problems that could arise from just that.
Yeah, that's true. But this is where like smoothness, like looking very closely at it, and can help.
Oscillation, variable oscillation.
Oh yeah, if I increase the input, it will change its behavior. This thing really wants to oscillate. Oh right, if I reduce this.
That's because of the very high Fy.
Yes. Yeah, sometimes it does weird things, but I think it's that square term now. Yeah, there you go. This is the normal winner-take-all network that we often work with, where a few things increase and then eventually the one that has the highest input gets picked. But then it's no longer sensitive to new input coming in. So you often have, so this is where Jesse and I we were talking about resets. Was that at the last session? A lot of the time people will ad hoc kind of just reset the whole network. I always found that really unsatisfying, and wherever possible, I try to have an intrinsic, because thinking about how a system can reset is itself an interesting question for just theoretically, right? Because you can come up with testable hypotheses for how they, like, how does an animal know what a trial is? They do all these interesting controls. They make the intertrial interval very, very variable, and but the animal has to somehow kind of add things in a way that t is equal to 0 is common across these different events.
I can give you a simple answer as to how an animal knows what a trial is.
Okay.
If he has got a drop of juice the last time, he knows that there is another trial coming.
But not when. But yeah. I mean, yeah. There's definitely, if events are, in fact, this is a point that Ram makes about looking in the time domain and interrupts. And a really interesting finding, which behaviorists found without actually looking at neurons, is that the delivery of a reward acts as a kind of reset. And I actually was reading a behaviorist paper, which is like, it's a completely different language. So I took weeks to understand what was in the paper, but when I understood it, I put that in the model I was making, and it immediately was able to reproduce the overall behavioral effect that I was looking for. So so the reward itself can cause the neurons that are active to learn and also shut them off, which is really interesting, sort of two kind of countervailing tendencies with the same reward. I'm not going to call it a dopamine signal because it's not necessarily dopamine, but it could be, and there are reasons to associate it with dopamine.
By the way, I just want to point out that in this winner-take-all in higher dimensions that Johan showed, while if you adhere to certain properties of like the parameter regime, you're always guaranteed for a winner. But it's not like oscillations are like some random idea. It's also been shown that these systems can exhibit oscillations if you have not set up your system properly. Like, you have to have your A and B and whatever, these Xmax and Xmin and all that stuff work in a certain way. Then you will have a winner take all. Otherwise,
and that's in the 73 paper.
Another thing is also the inputs.
And the input. So
in this case, the input stays on and it's only an excitatory input.
Yes.
Whereas the UMAP winner take all type things, when you have feedback, shut off the input at some point. So at some point, your input instills a pattern, you shut off, and then the feedback takes over, and you get the winner take all or squashing or linear memorization.
So it's not like this is reporting something like a new behavior.
Oh yeah, none of this is new.
These systems can oscillate if you don't structure them properly. Or if you do structure them properly, you can also get oscillations if that's what you want.
Here I have two winners.
Yeah, the analysis actually gets a lot harder when your inputs are still on alongside the feedback, but then also if your inputs are time varying,
yes,
it becomes even messier.
Yeah.
And each structural sort of change like that just makes it more and more difficult to analyze properly.
Yeah. But having seen these kinds of oscillations in these shunting networks so often, I'm pretty sure that that's what this is. It has to do a lot with the decay and the specific parameters.
Yeah, you can definitely
not necessarily decay too. You can also get it because of various other things. One, in this case, one of the like here you have used like a common maximum for each neuron, right? Like everything is between 0 and -2 or whatever. You can actually arbitrarily have different neurons have different max and x-min. It can still guarantee a winner despite those things having different maximum and minimum. So it has no problem with that part. But then you can actually get oscillations for like these very high inputs and no inhibition, as Saurabh was saying, or like, it's constantly on or it's constantly changing. You can get oscillations. The next time you can type of oscillation you can get is, this is one of the most important things. I think we didn't discuss it here. In this entire competitive dynamics part, the most important idea is how symmetric the connection is. It has to adhere to symmetry. In this case, these 20 neurons are such that 90 neurons are feeding back or whatever, the Gaussian symmetry is preserved or something like that. But in general, if there is no symmetry, there is no guarantee for you for a simple winner take all or simple uniformization or any other stuff. You can get oscillatory patterns. You can also get chaotic patterns. So all of that is possible, especially if you don't adhere to symmetry in the competition. It can cause chaos if you want, or oscillations.
In fact, the example you have above with the winner take all, I simplified that to just have some input that stays on, and then you shut it off, and then you observe the winner take all versus pattern preservation behaviors. And in those cases, the space of things to explore becomes much more restricted and less possibility of getting any unexpected behavior out.
Where's the fun without unexpected behavior?
It's a different beast altogether, right?
But yeah, all these weird effects, they're always lurking, and very often we want something stable. But I've increasingly been thinking about how systems get out of a stable state. So it's a quasi-stable state. So for exploration, it's important to be able to not stick to a winner. So you can end up with these highly biased situations. Like, let's say that you're exploring a region and there's a few rewards in some corner. If you keep just going there because those were where your first few rewards were, then you won't discover that there were better rewards or more rewards elsewhere in the room. But constantly exploring is also a suboptimal strategy. You'll find you'll have perfect representations of the probability of rewards in a stable environment, or even you'll be able to track even in an unstable environment, given the learning rate is fast enough, but you won't maximize your rewards. So switching between a winner take all that's based on prior learning and something that basically raises the temperature. This is the concept that they have in the machine learning world. They call it, because it's like the equivalent of temperature in a Boltzmann distribution. So that you can get something other than the winner to occasionally win. And people haven't necessarily modeled that, but there's interesting ways you can do that. I was doing that with the basal ganglia network recently. So once you have these little units, the little Lego blocks. So these are like networks that don't have a sense of space. Once you add the idea that the systems are not just inhibiting anything in the network, then you have these, we call them kernels, which are like the way that things interact nearby. And versions of this type of thing, they're not unique to anything that Grossberg did, because computer vision people use simple version, like more sort of algorithmically efficient versions of this for doing edge detection and stuff like that, without any claim to be neural.
So can you see my screen?
Ah, lovely.
So this is just an example applying the same winner-take-all idea to images. You can do this. So I mean, now everyone has like your filters in your photos and photo editors in your camera and all that stuff, so you can do it easily. So this is like the neural network version of the same. I'm not done. I mean, this is a very simple simulation. I could have run it for 1,000 more steps or whatever. So this is just the original image, and the linear whatever activation function is mostly the same, because it's going to preserve the pattern, but if there is any noise, it's going to increase. And the slower than linear part, you can kind of see that it's kind of whitening to some value. If I run it for a longer time, it's probably all going to become white and smudged and all that, but you can kind of start seeing that it's already getting smudged. The faster than linear, actually, I was surprised. In this case, it converged really faster. You kind of only see like a few dots of white patches on the image, which are like the brightest spots in the image. And the sigmoid is kind of like the best of all worlds. So it kind of like removes certain components and then amplifies some, all that. So this is kind of how we apply to the visual part in a spatial sense. But you can also run it for other modalities. And I think sort of Jesse and all, they've all done it in other modalities.
So one way you can think about what the inhibition is doing is subtracting out some kind of average. It doesn't have to be an average. It may be something like an average of a nonlinear sum, for instance. And so edges you can think about as in terms of like what's left over when what exceeds the average or what's not average. So Schmidhuber, I really like his this term he uses. Interestingness is the first derivative of beauty. And now that we have been talking about derivatives so much, we can kind of get a sense of this both in time and in space. So what is a spatial derivative? Like a spatial derivative will give you where things change. So the highest values of a spatial derivative will give you the edges. And these kinds of difference of Gaussian, effectively that's what they do. The difference Gaussian, depending on the size of it, the relative size of the excitatory and inhibitory Gaussians will kind of give you a measure of what the features you're interested in an image are. Like you want to look for a blob of a particular size and no bigger and no smaller. And it will pick edges of that size or things of interest of a particular size. So you can, and in a sense, that's what's interesting, because did I say this last time? So someone once asked what the phase component of a Fourier transform is doing. And I was very surprised because I suspect that what this person was doing a fast Fourier transform of a time sliced version of a signal, not realizing that they've actually put phase information in there and they already are using it. Because if you take, say, a sound signal or a picture and look at just the phase component of a Fourier transform, you'll be able to recognize the picture or the sound. What that means is that the overall intensities of different frequencies, either in the space domain or the sound domain, they give you some amount of information, but the main distinguishing information is all sitting in the edges. So competition, if we understand it as contrast enhancement, can be used, like you can just use it everywhere pretty much, and for any kind of object processing, you kind of need it. And it's not the case that the brain always does it in the way that it's explored here. The mechanisms might differ by a fair amount, but it gives you a good kind of starting point, because this is pretty much the simplest winner-take-all system you can make.
I think also the one good thing about that, like the steps you go through to determine, how that network's going to behave, it's really a cute method of analyzing dynamical systems in general, because Steve's like the thing Steve did was to say, look, if you process, if this is your dynamical system, it's processing some input, you can see actually that the network, the system is responding to a pattern, right? And that pattern is the ratio of the individual cell's activity over everybody's, and he shows how it falls out. It gets you thinking about dynamical systems in a way. It's just a unique way of analyzing what that network's behavior is when it would otherwise be too complex to understand, I think. Like the UMAP and 73 papers both do a really good job of that breakdown.
Yeah, the whole pattern energy factorization reduces the complexity of like dealing with cubics and whatnot, and like simply talking about where it's easier to just break it down into regions of where it is going to be monotonically increasing or exponential or flat. So it's much more easier to say whether the system will have like fixed points versus stable versus unstable. Like you can do a lot of all the dynamical systems analysis, but without having to worry about the fact that you can't visualize the phase plane or all that stuff, especially once the dimensions or the number of neurons are like more than 4 or whatever. So I think
even the sum of j not equal to I other terms.
Yeah.
And then as you're like calculating the steady state values or whatever, and you're just like, oh, that sums up nicely with the xi's activity, and then you have the total activation across the layer and so on. It's nice.
It's a funny little trick, but it's so satisfying. I'm going to just share that again just to highlight it. Because it's all it really is a trick, right?
Yeah, like if you do it even with just the bottom-up inputs and ignore the feedback cases.
Yeah, this just the fact that your inhibition is the other inputs, right, is what gives you this nice term of this total being this constant, the overall input. And it has a good interpretation physiologically that cells, even acting via interneurons, typically are not going to, or often are not going to inhibit themselves all that much in certain situations. So you could get this very nice normalization just from this funny little trick. You won't even see, like if you look at an equation like this, you won't even see necessarily that it is normalizing until you work through it. And it's strange that people don't use this equation more often.
It's not just the normalization of the activity, right? It's doing two things simultaneously. It's not simply normalizing the input pattern to be reflected. It is also a way to overcome, like it's a way to do proper rescaling of the input as well, in the sense that given that you and I will not have any luxury of knowing which condition you're going to be in. Let's say if you're taking the visual world, I might be in a nicely lit environment over here versus a very bright environment versus a very dark environment. You require this whole system to kind of go through these different input regimes and still work well. And I think it's kind of like does a double work of both pattern factorization as well as gain control in just one step. So it's effectively useful that way.
It's like when you step out into a blinding bright light, your eyes quickly adjust and you're able to see contrasts, and then step into a dark room and then quickly. So you're rescaling. You can experience that happening in the span of like a little bit longer than reaction time. So it's like factoring out the common factor. If everything has an overall brightness, then you can subtract that out. And that's what that inhibition does. But divisively, so that you always have the relative contrast.
I wanted to put this winner-take-all system into more context of how people use it. So the example we've been using is maybe neuron types or neuron populations or anatomical regions. And if you had two, you could have two groups or two locations. And then in your multidimensional, you could have multiple networks that have all exactly the same winner-take-all network connecting each one, right? Analogous to that, could you also have, instead of regions or cell types, you could have something slightly more abstract, like a state or a decision that you're deciding between course of action A and course of action B. For the two neuron model might be action A versus action B, and eventually you go with one, and that's the winner. Is that something people think about in that way?
Let me pull out Paul Chisek's work. Hopefully it'll be a good diagram. So Paul Chisek worked with Steve and my advisor Dan Bullock on evidence accumulation models. So the evidence accumulation can be thought about fairly abstractly, without having to say that it's a neuron or a group of neurons. But typically we interpret it as that. So the classic example, the studies that he was working in when he went to Montreal, were these random dot patterns where you have a screen and some of the dots are moving left and some are moving right, and you can in classic psychophysics style, you can titrate difficulty so that, and the animal has to pick which is the net direction in which the majority of the dots are moving. And so you can think about that in terms of, so the two inputs that are going into these two. Imagine that there's a node for the button that says left and the button that says right. And the weighing scale is like going, and eventually a decision is made. What's interesting is he showed that there's an additional factor. The animals are not just accumulating evidence. There's also a kind of urgency signal. So you can add that into the, well, it could just be this feedback, because that feedback will mean that once something reaches a certain level, intrinsically it will just pick a winner. With the when you pick, especially if you use a sinusoidal, a sigmoidal transfer function, but it seems more likely that's a more broad network effect, because it can be modulated by hunger and I think urgency signal is the perfect name for it, right? Because it could be anything that causes the animal to want to make a decision based on, well, I don't care anymore, I'm going to decide, I don't have time to wait. But
also at the hand waviest level, because a lot of these models motivated some of the more machine learning-y versions of them that came out, like the fuzzy art map type stuff. You could think even at the level of like object recognition, you want to have a single node that becomes active or whatever, or in the course of some incoming sequence of, let's say, sounds, you want to be able to recognize a word, and having competition between potentially representative, like objects in the vision case or words in the sound case. You have a sort of winner-take-all network that selects the correct output. But again, the direct linkage between the sort of more biologically plausible versus the more machine learning cases gets it's hokey at points. Involve like fuzzy logic, and then there's some hand waving that goes on in terms of like, let's make it work on the machine learning side.
3x2 rule, 2x3 rule.
That Jesse has got all this working, by the way, guys. He's got an art-like system with a completely neurobiologically plausible, which is as good as we can get, right? Because very few people are going to be able to test it, let alone willing to test it. So for now, I think.
I mean, I was promoting it a little bit, but it does some of these things. It does some of these things. It is a little wonky and not always stable. But.
What's the best part? You didn't talk about it. You should actually say like how you got it to like work on. I don't think you published it yet. The whole like alphabet and like numerals detection, like visual, like MNIST data or whatever that you were trying on. That's probably.
Yeah, I didn't I think I might have tried on MNIST at some point, but I did try on like my own created alphabet letters where it can memorize 26 letters from one single presentation. And yeah, but anyways.
But Kale, there is another application that you can think of which is not restricted to neuroscience with these kinds of networks. You can just assume that each of these X's are different animals in an ecosystem. Okay. And you can run a full-on predator and prey model for multiple species in an ecological setting. Let's say that you have some initial values for different species in your area. Given that it's you can fix some arbitrary threshold to say that if it goes below a certain value, 0.001 or something, you can say that the animal goes extinct. You have to make that alone an extra step. But once you do that, you can kind of like go, you can actually see the dynamics of like how species are proliferating and like, we're getting control from one another and all that. So you can use the exact same model that is over here, just increase the number of, have to have like a minimum and maximum, like Johan had with Xmax and Xmin to be the same. You can actually have different Xmaxes and different Xmin, and the system will still give you some dynamical result. So it's not restricted to just like 0 to 1 or whatever. You can just have arbitrary population values or percentages, and you can do this. So it is also a generalized competitive system for anywhere you want to talk about competition. If the only thing,
you can even use it for like traffic routing, right? Like when you have to, there are multiple paths that you want for redundancy for routing traffic, and based on traffic bandwidth, send your traffic through a channel that is not very saturated or things like that. You can use things like this to like dynamically drive traffic, select traffic paths.
In fact, even Steve's like post neuroscience attempts at becoming pop famous were to use the shunting equation as a metaphor or model, or I don't even know how in depth he got into it, but of the economy. And you could think of the signal functions, those feedback functions, as potentially rewarding various types of levels of actor in the economy, right? So if you have a faster than linear feedback signal, that means the more money you make, the easier it is to make more money, right? So the higher up on that scale you go, the faster and faster you're making money. That makes it easier to inhibit everybody else in the economy, and they're going to quickly go down, and all of a sudden you have a winner-take-all network, right? And there's actually interesting like overlaps with Piketty and R greater than G and blah, blah, blah. But then the slower than linear signal says that as your X value goes up and up, if you had a slower than linear signal, the feedback that comes into you is actually going to reduce and reduce until, it's effectively the same as whatever other people are getting. And in those cases, there's a uniformization tendency where people will get sort of squashed into a more narrow region of the space, more similar to linear signal function basically just preserves patterns. So if you start with a certain amount of money and certain ratio in the population of wealth, it stays that way. And so it's an interesting way of thinking of those signal functions.
I mean, in general, you can make the case that this is, whatever your opinions of Steve or not, the competitive dynamics part is, I would say, the most generalized competitive dynamics that you can find anywhere for any number of variables you can think of. So if you want a very good competitive dynamics model, this is probably the best end you will ever get, unless you're developing your own new technique. I don't see anything that's even remotely better than this when it comes to competitive dynamics. But if you want to do other types of dynamics like add cooperation, then it's a whole different ballgame. It's a very big problem that these systems can't deal with.
That was actually my next question, was for these two neurons, right? The relationship between them that you drew and that these equations represent is the winner-take-all system. But I would imagine you could have a whole collection of different little motifs, I don't know what you call them, of the microcircuits, and you could have a whole zoo of those microcircuits. Is there a review that presents those little microcircuits?
Yes, there is no review necessarily, but I will put it this way. What we see for 2 and 3 dimensions, you shouldn't immediately and just translate it to work for higher numbers. Okay, so you can actually start seeing a lot of interesting patterns if you relax certain structures that are inbuilt into this network. That's what I meant by the symmetric competition part of it. If you relax the symmetry even a little bit, you can actually get really, really quirky behaviors, not just oscillations. You'll get extremely quirky behaviors in high dimension, such that it could be possible that there might be, let's say that there are 10 species or 10 whatever neurons, whichever way you want to think about it. Five species are actually behaving, if you look at their phase space, they're probably behaving like they're in cooperation with one another. But those 5 could be in complete competition with the other. So it's kind of like even forming clustering and all that stuff. So this symmetry part was just enforced because they were able to prove certain very global theorems about stability and all that stuff. But if you relax it, you actually get way more interesting behavior. Like you could like see like in just 2 or 3 guys forming a cluster and being cooperative, and then they're competing with other guys and so on.
Those weird GIFs that I made are basically that. I added just one other thing to keep it dynamic. So the zoo thing was partly, so over the past few days, I've been playing with just this two competitor system just to look at it. And once I got the basics, so this one on the right is the basic kind of winner-take-all system, where you have a stable attractor here, you have a stable attractor here, you have some actually weird stuff happening, sometimes stable, sometimes unstable.
Can you make them a little bigger, Johan?
What?
Can you make them a little bigger in some way?
I don't know how to, like I tried full screen and then it kind of shrank again.
Okay.
Okay. See, it just bounced back to,
okay, yeah.
Wait, no. This photo viewer is weird.
The plus sign, the plus symbol at the top by the trash can.
Okay. What? Oh, okay. Is that better?
Yeah, thanks.
And so then I just sort of changed those functions, F and G, which we talked about, which are the most interesting aspect of these systems. And I found that it doesn't have to be a winner-take-all system. You can get another sort of interesting discrete system with attractors. So you can't really see it here, but where these null-clines meet, there's an attractor, there's another one there. And there's an unstable point here. And then there's another attractor up here. So this is sort of a somewhat cooperative system, which is that when there are big differences, one of them wins. And when the differences are not too small, which is you can actually control how large this space is by changing some of those parameters, then they both get to be active. And I don't know whether this is useful or anything, but I just thought it looked pretty cool to see all these attractors and unstable points in a system that we've been working with for a decade and a half. So yeah, that was just RNNs, recreational neural networks. But
what did you change to get that? I'm curious.
So so instead of both of them being faster than linear, I made the inhibition sigmoid and the excitation
faster than
linear. So, and then I really amped up the excitation. So
basically we can do that for 4x4 matrix that I was telling about. Like we start getting a zoo of, even for 2D, you'll get a zoo. But the problem is you should be very careful when you, like I said, when you extend it to more than 2D, you have to be careful about other global structures. The symmetric competition is a global structure. And that was like the main theorem from Steve and Mike Cohen. They had to have that, otherwise they couldn't do certain things. In fact, if you read the last paragraph of their paper, it's kind of like a sad lament, and it's true to this day, that they were saying like, we can solve everything about the competitive dynamics world. But if we start introducing any cooperation into our system, we have no clue about how to think about our system and how well it behaves. All we can do is just have simulations which kind of work well, but we can't really prove any good theorems about like whether the system would be stable and all that stuff. And this has huge implications because it matters to society. They had all these lines in that last paragraph of that
I
paper.
don't remember this at all.
So when
Interesting.
you
I have
so
no
when you say
recollection.
so when you say symmetry, you don't mean symmetry like spatially. You mean symmetry between the excitation and inhibition,
No, symmetry in terms of the connectivity.
like spatially.
Spatial, not like, yeah, you can imagine them to be spatially, right? If you have, imagine like n neurons or n nodes, the way in which they are connected is such that it has to be, the matrix has to be symmetric. So if A is connected to B, B should also be connected to A.
Okay.
If that order is not followed, if there is no more symmetry, you can actually start getting weird behaviors.
So like this is one of those weird behaviors.
Is
This is
this
like-
actually, Karthik, is this, I wanted to mention also that they do a shout out to voting systems, various types
So
of
that's
voting
the same,
systems.
that's the point I'm trying to raise. That is
Yeah,
the
Kale, I
example
think this is something you should check out because you're into.
that is the example that they give to show for just a 3D system where there is no symmetry. The Leonard May voting model, which they converted, it's the old Condorcet problem. I think we talked about it briefly, Kale, sometime back. You have no way of picking your ideal winner because it's kind of like going in a circle. So that's not a good competitive system. So that's where they use that example as a motivation to say why the symmetry matters. And in fact, not just Steve, right after Steve did this in 1973-74, May and Leonard did the voting model for just 3D. And then Stephen Schmale did it for ND, and he showed that there is a whole zoo that will happen, zoo of possibilities in terms of quirky behavior and all that stuff. And you can take the Cohen-Grossberg world as like saying, like, if we restrict that zoo of behavior that Schmale has shown and have some structure in place like symmetry, blah, 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 monotonicity, all that stuff, we can guarantee you stable global competitive dynamics of n equal to arbitrary number. I mean, that's an insane thing, right? Like, imagine like there are 1,000 agents doing whatever the heck they want. As long as there is the symmetry thing that is adhered to, it will actually be always stable. The system is not going to go blow up. I mean, it's quite insane that, you know, they can like not visualize, do none of those things, but kind of like have a theorem that can show that. It's
life is not stable, right? So, and nor is the brain. So it's not always clear to me why people want things to be stable. I mean, it's good to know that things can be stable, but
they came from a different worldview, right? Because they are looking at pattern formation. For them, pattern formation was the goal in these kinds of systems. If you're going to have a pattern formation system which is going to be very unstable, then what are you really storing? Because this is kind of like the step for them to go into categorical learning and art and all that stuff. So they kind of want, at least at one level, some network will faithfully render some pattern into something useful that you can then go forward and do whatever you want. So it's the one place that they kind of want some sort of an isomorphism with the world that they can then move up and abstract. So they had to come up with these structures in place. But as I said, if you're not interested in the symmetry, remove the symmetry part, tweak it even slightly, you will start seeing extremely quirky behavior. I mean, this is one example of it, like faster than linear, and then, you know, sigmoidal learning on the other side. So you'll start seeing.
What I want to do is just have like where the on center and the off surround are not centered on the same points. So you'll get like instead of those Gaussians that he showed over there. Oops, I scrolled down. Instead of it's in the other quick. In here. So instead of this, like just having the centers offset, what happens then? I'm trying to try this at some point.
You might be interested in some of the models that people have made about ring attractors.
The ring attractors, yeah, that's what I figured as well.
And there's a model by a group in Brandeis, actually a spiking model ring attractors that
Works like that.
yeah.
And I imagine that in a sort of where there's no closure, you can get like sort of sending out patterns, right? Like you like constantly churning out kind of. In fact, these types. So the cooperation, these bumps that can stabilize themselves is not an example of cooperation.
I mean, you can say it is cooperation, but in the sense that they didn't actually add explicit cooperative rules in the system. So that's what I meant, like subpopulations of the system can cooperate, but the overall system is not set up for cooperation. So you might have competitive dynamics all the way through, but yet you can see subspecies or subspaces where there is cooperation. So it's sort of like, you can make the case that you can, if you want to get cooperation in your networks on subspaces, you don't actually need explicit cooperation. You can kind of get it where some of them will be cooperating and some will be competing with them. But
how do you consider like the bipole stuff? Is that cooperative dynamics, or is that still in the competitive frame?
You mean the dipole, the dipole model?
The bipole model.
You mean the cells, that the visual cells?
Yeah.
I would say they are more, they're still competitive actually. The only difference is there is a cooperative kernel. So that's where it's all simulation stuff. It's not really like, we don't know the actual, how the dynamical system behaves. It works well, but we don't know whether it is true for any regime, all that stuff. So it's not analyzed in the same way that this kind of system has been analyzed. So it has cooperation. In fact, all our models have those cooperative kernels in our visual system, but that's only because we are doing simulation models. We are not doing the studying the whole dynamical system as such.
So if you move to the landscape model, I guess what the stability is something like this: that if I have a group of neurons and I want to learn categories of, say, things, objects in the world, I'm creating valleys that are deep enough and well separated enough that I don't confuse cats with dogs, right? And because it takes time for these things to form, I don't want an existing valley to be hijacked by some occasional weird experience and at the same time, I want to be able to form new categories that are, so I think in Steve's case, he just adds new neurons, right? Like in order to deal with things that are radically unlike anything else. So for the brain, we don't add new neurons, but we just have uncommitted neurons that sort of become representative of some new category.
My favorite example with cats and dogs is hyenas, which people think are dogs, but are actually ancestrally more cats. That would, if you had a simple like winner take all, you're trying to decide, is this a cat or a dog, and you build up evidence, you're first going to get over into the dog, but you would need some huge bump to get back to.
In fact, that type of learning is not well modeled at all by anything in dynamical systems, because it's semantic learning, right? So you don't need to be told millions of times that a hyena is more related to a cat than a dog, right? So it's not the kind of thing that like, well, does art have one-shot learning? Not really, right?
No.
One-shot learning is really, really unstable. In all the networks that I've seen.
Actually thinks one-shot learning can't work.
Yeah. I've tried to do one-shot learning and seen it just explode.
Wait, doesn't it have one-shot learning? I don't understand how you're defining one-shot learning.
I'm not sure. I don't think Steve
does, right? I mean, you give it one stimuli and it's.
Oh, right. I suppose, yeah, you're presenting it for long enough that it doesn't need to be shown repeatedly.
Right.
I'm not sure Steve thinks there is one-shot learning. I might be wrong though.
Well, in hippocampus, like for instance, you can have a place cell forming after one stimulation even. So you can get the cell to learn a place just by a single stimulation.
Now, are flashbulb memories one shot?
Yeah, that's a classic
That
example.
is, we all know exactly where we were on September 11th, right? When we first heard the news. No, none of us will ever forget that.
Yeah,
It's getting hazier, so I don't know.
January 6th, right? That's one shot learning.
That's one shot learning. Yeah. But so I guess for art, in the sense, well, it's weird. It's a weird thing because it's not like Steve's models are not for episodic memory. So one-shot learning is typically episodic memory, right?
Right. Right.
So you have exemplars of, like, you see different dogs, and you need to see multiple dogs in order to form a representation of dogs, right? For in art. So that's what I mean by one. So in one-shot learning, you have a representation of the exemplar and not a category.
You could, in principle, do that with the in-star, out-star rules and just set the
right. Speed up the learning rate.
Yeah, essentially,
and
get it to converge to whatever the input pattern was, or whatever however you represent the episode.
Yeah.
And usually, I don't know how easy it is to learn a whole category in one shot, right? And
impossible.
or something that you have not already in a way learned. Like, can somebody, for example, learn a I'm trying to come up with a good example here of
all subgenres of a music you have not heard yet.
Yeah.
I mean, of course, you can't learn something you haven't been exposed to.
Like, if I were to play you one example of all the different.
No. So yeah, or something like a language, right? Let's say there's a language I've never been exposed to, and can I learn a passage of that language through one shot? Some like episodic sense. Like I had some very, let's say, significant episodic event, right? Can I just learn the passage of an unknown language?
That's a good
definitely.
example.
No, but how about learning name of a person?
If it is, let's say, if it's in a language you do not know, right?
And pronunciation is very difficult.
No.
Yeah, and pronounce, like if I go to, like, for example, say, what's a language I don't know, like Finnish language, right? And it's the name of a place in Finland. Can I learn that through one shot?
But that's different. That's because that's different. That's because you have to learn the parts before you learn the actual thing, which is a whole nother.
And arguably, you kind of can.
You're not learning one thing. You're learning multiple things, multiple levels of the hierarchy, and that's why you can't learn it at one shot. But you can still learn one thing.
That's that's a point, right? There's no such thing as a single thing.
No, you don't have to know. No, you don't have to know the language. You can equate it to some known syllables in your language that you know, or your mother tongue, and you can reproduce it.
I doubt that you can reproduce it, but I think you might have an episodic memory. But it probably, I suspect even the episodic memory will be vague.
I think
if someone shouts a foreign word at me, and before that bangs the drums, and it's so it's all significant. It's not like I'm necessarily going to. I'll remember that it happened, and I might remember some aspects. But I'm pretty sure that if it was in a language that I knew, that it would be a better memory.
Yeah, like if I don't know music, right? Then no amount of episodic learning can help me remember what chord was played, for example, during that time, right? So I won't be able to say, oh, that's a, they played a C major or whatever.
Ton of nuance here, right?
Yeah, this doesn't really have to do with one-shot learning. This has to do with like.
Jesse, what's a good example of one-shot learning then?
Like being able to, for example, like you can show someone. Well, yeah, learning someone's name is an example, I guess.
If I show you.
But also, like, you can show someone like 1,000 pictures, right? And they can tell which pictures they've seen and which pictures they haven't seen from seeing the picture once, and even only presentation for like 500 milliseconds.
That's true. Yeah.
But the pictures need to be in a domain that they're familiar with. Like if I show someone pictures of fractals or let's say pictures of like cells, neuron cells in the brain, right? And they have no like anatomy background, right? They wouldn't be able to like remember. Actually, I saw a pyramidal cell.
So even there, it's nuanced. Okay. So experts have a very good ability to memorize. Let's say you show them an image from a radiology image, or a chess player, a chess setup. They're very good at remembering legitimate chess structures and legitimate images from radiology. If you make some weird changes, if I give you a random pattern on a board, a person who's not experienced in chess will have a better memory for those situations, right? Now, the problem with the language case is that there's also, you're parsing it in various ways, right? So you're going to map it onto linguistic structure that you already have.
Right.
Manju's point is that is always true for everything. And like, if you're going to make a generalization, you want to make, you want to say that about phonemes, you want to say that about, I mean, what is the story of?
But that's not relevant to one-shot learning. That's relevant to like hierarchical learning.
Hierarchical, so it's not just that dogs and cats are the categories. It's even things like ears, nose, texture of fur. All these things are categories too.
Agreed.
In the modern like way of understanding perception, right?
In the name case, it's obviously the case somebody can tell me a name from a different language that I've never heard, and I can memorize that name, right?
Well,
but
in one,
to Jesse's point, that's not necessarily one-shot learning per se. Like if I show you a drawing of a widget, and I don't tell you any specifications about here's how you distinguish widgets from, machines or computers and blah blah blah. I show you one example of that, and then we're going through a series of images. Being able to determine widgets from other things I've never seen before is the ability to do one-shot learning, right? Interestingly, people have started seeing if machine learning models can do zero-shot learning, right? So can you create categories for things that you've not been exposed to explicitly, right? So maybe by either explanation, you have to be, you're given a linguistic explanation, you have to be able to pick out from images. Or if I tell you, this is not the thing, this is not the thing, now go find me the new thing in the set. Those are cases even of zero-shot learning, right? So it's just all a very nuanced territory at that point too. Like the name case comes down to working memory as well, right? So some people can memorize a phone number in one shot, some people won't, and a lot of that has to do with how.
But let's say you've never
spoken. And some people can memorize a phone number from a specific country, but not from another country, right? Because it's like I had this problem in India. When I was in India, you give me a phone number, I'll memorize it, like almost one shot.
Well,
but then
there's add in the problem of cell phones ruining people's working memory for memory for phone numbers, but
like if you imagine that you've never heard a tonal language before, and then someone says, my name is and then you're like, and then you have to remember the name, like.
But there's a bunch of.
But that's because you don't have the pieces. Like you can't remember Shakespeare if you don't know any words of English. How are you going to remember Shakespeare if you don't know any words of English? It's an impossible task.
That's Himanshu's point.
No.
That
like wait. I don't know any words in Hindi, but I can memorize Hindi songs.
Yeah, you have to hear them more than once.
Right?
Yeah, try and remember like a whole
sometimes
play.
You remember at least the first, second line when you come out of the movie, you learn it, right? The first time you watch it, you learn the first line, no problem, you sing it.
Ram,
so that's what I'm saying, episodic and semantic, it's very difficult. I don't quite know where we are going with this one-shot thing. We need to first define what we mean by one-shot.
Ram, I'm very proud of you. Ram, I'm proud of you. You're a true South Indian. Like, I don't care about what Hindi is. I can just sing songs.
Same issue of the existence of common ground is there, because songs have melodic structure that transcends languages, and Indian languages have a lot of common syllables and morphemes. And also because of Sanskrit terminology, there's a lot of similar vocabulary also.
There's also a lot of interesting overlap with Sapir-Whorfy type hypothesis stuff with like color perception, right? So a lot of languages won't place labels on various types of colors. Actually, it's interesting, right? If you're a language that has only two color names, two color words, it's always black and white. If you have three color words, it's always black, white, and red.
Yes.
And then languages will start to expand out. But the memory you have for various types of color sequences you might be shown is affected by the labels that you've placed and where you've placed them, and your ability to pick out colors. All of that stuff is really like tied together in ways that it makes this a complex.
What are those
complex
two
web.
Russian words? Goluboy and something? The
Is
two
it like an
light
aqua
blue, dark
blue?
blue?
Yeah.
Boroditsky. Lira Boroditsky. He does tons of
Boroditsky's, yeah, huge on the Sapir-Whorf stuff. That's a whole separate topic, but a fascinating one.
Yeah. Anyway, I'm gonna stop the recording.